Good morning. My name is Gerard Kleywegt. First of all, I'd like to thank the organizers, Jose Maria and Elena, for inviting me to talk to you this morning about community recommendations on validating Cryo-EM models and data or maps. So first, a few slides about what have we learned about validation from crystallography mainly. Then what have wwPDB and EMDB been doing over the years in the area of EM validation. Then some of the recommendations from a meeting we had in early 2020 of the validation task force. And finally, some of the work we're doing at EMDB, partly implementing the recommendations, and also some other work that I'll discuss. So validation really is critical assessment of your model, and in case of Cryo-EM also your map. So just focusing on the model, your model should at least explain all the data that you used, and it should explain all the prior knowledge that you used, for example bond lengths and bond angles. But that just verifies whether your refinement program has done a decent job. In order to assess the accuracy and correctness of your model, it's much more important that it does some of these things. So, does it explain the data that you didn't use? Does your model explain any prior knowledge you didn't use? In general, is my model the best possible and most parsimonious explanation of the data? And if you can make predictions on basis of your model, can you go and test them in the lab, and are they borne out? And if all these questions are answered with no, you really do have a problem. So validation usually entails comparing your model to the experimental data and to the prior knowledge, and it should at the very least reproduce any information that you've used in the construction of your model. So you used your reflection data in the case of X-ray, you used bond length restraints, etc. But it should also predict any knowledge or data or information that you didn't use while constructing the model. So the Ramachandran plot, for example, the rotamericity of your side chains, the packing quality that your model displays. Those are things that are usually not restrained during refinement, and so they're good validation measures. But you have to be aware of Coulthard's law, and I'll explain in a minute what that entails. It's good to use global and local metrics. Global metrics to assess the overall quality, local metrics to look at the details. And it's good practice to assess the model alone, the data alone, and the fit of the model to the data. And so if your model, again, if it fails to do any of these things, you better have a very good explanation. So global model quality can be assessed with global coordinate-based statistics very often, but for individual residues, you get the result that a residue may or may not be an outlier. And an outlier can be one of two things. It can be an error in the model, which means you have to fix it, or it can be a genuine but unusual feature of the structure, and then you probably want to describe it in your paper. And the way to tell the difference is to look at the experimental data that you have, and hopefully you can tell the difference. And it's always good to keep in mind that extraordinary claims require extraordinary evidence. So if you find in your model example of chemistry that has never been seen before, you better have the experimental data to back that up. Now I mentioned Coulthard's law, and that says that when a measure becomes a target, it's no longer a good measure for validation. And so, for example, some programs allow you to restrain the torsion angle values of your main chain to lie in the favorable areas of the Ramachandran plot, but that means that your model will look more like a protein, but you can no longer use the Ramachandran plot for validation. And you have to keep in mind that the aim of validation is to assess the quality of your model and the data, and to identify the aspects that may require closer scrutiny, in our case often rebuilding. And the aim of structure determination is to produce the best model that you possibly can that explains all your data and fits the prior knowledge. The aim of structure determination is not to get zero Ramachandran outliers or any on any criterion. Now, as of 2021, EMDB has joined the Worldwide Protein Data Bank. So this organization now manages 3 archives, the PDB, the BMRB, and EMDB, and it's made up of these five members. wwPDB has convened validation task forces for X-ray in 2008, for NMR in 2009, and for EM in 2010, made up of community experts and they've given recommendations, and some of the core recommendations have been that we should use metrics, etc., that are reasonably well understood, easy to interpret, generally accepted in the community as being useful for validation purposes, and ideally have been in use for some time, so we have good experience and understand the strengths and the weaknesses. Those metrics should be implemented in the software pipeline, and the software pipeline should produce a human-readable report, as well as a machine-readable file with all the details. And the software pipeline should also be made available as a validation server. So this is what has happened a number of years ago already. From the EM point of view, there was a first validation meeting in 2010 at Rutgers, and the report appeared 2 years later in Structure. The principle was that models had to be validated in the same way as for X-ray and NMR, and at the time there was not a huge number of map and model validation methods available, and you have to realize that this was before the resolution revolution. So when in 2010 60% of the EM maps released that year was at worse than 10 angstrom resolution, for example, and that's the current archive is very different from that. And just to remind you where we were in 2010, there were less than 1,000 maps in EMDB. There were just a few hundred structures in the PDB, EM structures. We were in the era of blobology. These are some of the typical state-of-the-art structures from the first decade of the 21st century, typically at resolutions somewhere between 7 and 12 angstrom. Now, in 2010, Kathy Lawson, who will talk later, made a prediction based on extrapolation of how many entries there might be in EMDB in 2020 based on what we had in 2010. And she came up with the answer of about 10,000. And at the time this seemed ludicrous, but of course this was exactly borne out 10 years later, when last year we celebrated the first 10,000 entries in EMDB, and we're expecting that the next 10,000 entries will already be reached next year in 2022. So in 2019, the UK EM Validation Network then decided to organize a forward-looking workshop. Where is EM going? What are the unanswered questions for validation? What should we be focusing on? So we invited 50 people from all over the world and had 2 days of very good discussions on single particle map and model validation. These are the kinds of questions that we addressed in that meeting. It was supposed to be an entirely forward-looking meeting, but of course we also discussed what should be in table 1 in papers, for example, and what should be deposited in PDB and EMDB. But it was a very useful meeting nonetheless, and there's a white paper in preparation. Then, given all that had happened since 2010, it was high time for a second validation task force meeting for EM. So this happened in January 2020, just before the first lockdown. You may recognize 3 of today's speakers as well. So there's Greg, there's Rangana, and there's Kathy, who also were at this meeting at the EBI. So in a couple of slides, I'll summarize the main recommendations and outcomes of this meeting. There were recommendations about how wwPDB and EMDB could improve data and metadata capture. For example, demanding that models be deposited in MMCIF format, perhaps capturing particle picking metadata. Half maps should be deposited if they were used in the study, and there were also recommendations on how to group related entries in the PDB and EMDB. At the time, there were some rudimentary EM validation reports available, so we got a lot of comments and recommendations on those. And there were a few recommendations on pure model validation. And one was to track what model restraints have been used. This is to figure out if, for example, Ramachandran restraints were used or not. And also related to that, to come up with an additional coordinate-based metric that is not biased by torsioning or restraints, like, for example, Kablam in more probity or a packing score or something like that. With respect to data and map validation, we got recommendations to evaluate a number of programs that assess local resolution, and also programs that look at the map anisotropy and angular coverage of the particles. There was no clear favorite in either of these areas, so the recommendation is to implement a number of them and run them on the entire EMDB archive and see how well they perform. There was the recommendation to add symmetry analysis, and there's a program from Garib Musudov's group, ProShade, which does that. And there was also, it was noted that deposition of particle stacks would be extremely helpful for validation and also for reprocessing and maybe re-refinement. In terms of the validation of map and model fits, it was noted that there was no good metric in the current validation reports, because we have atom inclusion there, but that's very subjective, because if you change the contour level, of course the number of atoms that are included inside the map increases as well. Again, there was a recommendation to evaluate a number of different statistics, EM ringer, Q-score, SMOC, etc. And also to add map model FSC plots and see how useful they could be. We were encouraged to evaluate methods for different map calculations, and also an interesting suggestion to visually illustrate map model fit both in a relatively good area of the map and in a relatively poor part of the map. So EMDB particularly was asked to implement many of these methods where no single program can yet be recommended, so that the whole archive can be run through these programs and analyzed, and also experts can look at how these metrics perform on individual cases that they are familiar with. There are also a number of challenges for methods and software developers, for example, to come up with criteria that could go into these so-called slider plots, the summary plots in the validation reports, that would have to be hard to fudge. So not things like the atom inclusion score. There were open questions like, can 2D raw data, particle stack, be used to validate 3D maps, and how? There's a need for more model validation criteria, particularly for reduced models, for example, C-alpha only models. It would be fantastic if we had a method that could define an unbiased contour level, both globally and locally. And it would also be very helpful if there were methods to assess if structural features observed in a given map are commensurate with what you expect for that resolution that the map is at. So the follow-up for this the meeting and the recommendations is that we should of course publish a white paper, so that's in preparation. And we've done a lot of implementation already of the easy to do recommendations. They have been added to the OneLab validation pipeline and the validation server. And also the validation reports for all EMDB and PDB entries have been updated and released. And we're still working on this second part to add a lot more validation checks to the web pages. At a later stage, more of these methods will make their way into the validation pipeline. We also have to wait for the field to do more work, and in a few years' time we have to go back and review some of these areas. And the meeting focused on single particle only, so we also need recommendations specifically for other EM modalities like helical reconstructions or electron crystallography. And then of course there are always unexpected developments and new challenges. For example, recently we released AlphaFold DB, and that of course immediately gave people the idea that, wait a minute, there are all these old low resolution maps lying around, and now we can go to AlphaFold DB and see models that look as if they fit those old maps. So why don't we go and put those models in those maps and refine them? And of course, validation at that kind of resolution is a whole new challenge. One thing we also did was to organize a symposium last year in November, which was about validation and also application of Cryo-EM to SARS-CoV-2. if you're interested in some of the presentations are available on this web page. Now EMDB has taken the advice and the recommendations from the validation task force and began to implement them. So we now have a 3-tiered strategy on how to do validation. Tier 1 is the validation analysis pages, and this is where we try out lots of different things, implement loads of different programs so that specialists can look at them, so that we can run analysis over the entire archive, etc. So we call this the validation analysis pages. Then on the EMDB website we have the validation tabs, and they contain a subset of the metrics and plots, etc. from the validation analysis pages. These are usually things that are tried and tested and reasonably well understood. And finally in tier 3, after a while, when there's consensus in the community and among the wwPDB partners, things can be moved into the validation pipeline that wwPDB uses to validate EMDB maps and PDB models. So I'll run you through a couple of validation components that we now have on one or more of these tiers, and I'm using this particular example, this entry, if you want to look it up. So it's a reasonably well resolved entry. It has half maps, so we can calculate raw maps, and it's a SARS-CoV-2 spike protein structure. So tier 1 is these validation analysis pages that you can get from the EMDB website for every EMDB entry. Tier 2 is the validation tab on the EMDB entry pages, so that's a reduced subset of the validation analysis page. And tier 3 is the validation reports, the PDF files that you can get from the wwPDB archive and all the member websites. Now, some of the new things we've done is we've added new projections, maximum value projections and standard deviation projections. And we also started to experiment with false color there. So we think that these projections help to assess whether you see features, particularly helical features, in a map that you would like to see, especially if there's a model. So this is available in tier 1 and 2, and the grayscale images are also available in the validation report. So that's tier 3. If there are half maps, we calculate a raw map. And again, we show a number of slices and projections in all the 3 tiers. We also show the view of the map. The contour level is not defined, but what we do is we set it such that the volume enclosed by the raw map is the same as the volume enclosed by the primary map. So it's approximate, but it works quite well in most cases. Then there's a bunch of plots related to the primary and the raw map. So there's the voxel value distribution, volume estimates, rotationally average power spectrum, and FSC curves. We have a number of views and plots that show how well the model fits the data. So there's the visual views, and there's an atom inclusion graph, and also in tier 1 the map model FSC. Atom inclusion is included also in all 3 tiers, so it's not so useful because it can be fudged with selecting your contour level a bit lower than you might normally do. So what this does is the percentage of atoms per residue or per chain that fits inside the map given the recommended contour level. Now a newer and better method probably is the Q-score, and there will be a talk by Greg Pinterley in this session, so I won't go into any of the details. For the moment that's only in tier 1, so that's all experimental. Also in tier 1 is 3D Strudel, which is something we have developed ourselves in collaboration with Gerard's lab. What it basically is model-dependent map feature validation using resolution-dependent map motifs for all the 155 Richardson rotamers of the common amino acids. So we check if you build something as a tryptophan, does the map around that residue look like a tryptophan map at that particular resolution? And if not, what does it look like then? I mentioned the symmetry, so ProShade has been implemented, so that can be used for sanity checking, applied symmetry, but it may also reveal approximate symmetry or symmetry that was missed in the map reconstruction. A much requested feature is to show how the model fits in the map, not just for the asymmetric unit of a virus, but also to show how the entire model of the virus fits inside the map. So that's been implemented also in all three. I mentioned 3D Strudel, so I'll briefly explain what this method does and give some examples of its application. So the idea behind this is to derive typical map features of all the 155 rotamers of the 20 common amino acids in EM maps in various resolution bands, and then to use these little maps, we call them motifs, to validate maps that people are working on. And so we selected a bunch of structures, we re-refined them with RefMac, and then used a number of validation criteria to select what we thought were good map and model combinations, and we used those to pick up all the instances of all the 155 rotamers and to extract the map around those residues and average them. So that gives us per resolution band a library of 155 little motif maps. Now those you can then use in a number of ways. The way we have been using it is, well, if this is a map feature and you have modeled it as a phenylalanine, does this local map look like you would expect a phenylalanine map to look at this resolution? And so this has been implemented, and there is a very nice ChimeraX plugin called Strudel Score, which can be used to look at the results of the calculation. So it shows, if you click on a particular residue, it will show the model and the local map, and then on the right it will show each of the 20 amino acids, how well the best fitting rotamer for each amino acid fits this local density. And so this is a nice example. It's a histidine, and the map really does look like a histidine. The only other thing that comes close is a phenylalanine. So it's a very reasonable interpretation of this map. We can also show other residues, how their best fitting rotamer would look like in terms of the map. So the experimental map is always shown as a chicken wire contour. A green map means that this is the motif that belongs to the residue that was built in the model. So here it's a histidine. This is what a histidine looks like on average at this resolution. A red map shows the map for a residue type that fits better than the one that was modeled, and the grey ones are all the other amino acid types. So you can look at any amino acid type in any position in the model. Now the last slide, this is just an example. This is a SARS-CoV-2 structure, an early one, where both we but also others all independently noticed that there was something odd going on in the C-terminus of a particular chain, and we figured out, and others and the authors as well, that there was a register error there by about 9 residues, and it was very easy to see in the Strudel plot. So you can see here that there's a number of residues where the green dots are really considerably lower in value than the well-fitting ones, and that there are red dots. That means that there are other residue types that would fit better in that position. So for example, this density feature has been modeled as a leucine, but obviously we can tell, or Strudel can tell, that a tryptophan or a tyrosine or a histidine, something big like that, would fit much better here. And indeed, in the corrected model, it is a tryptophan. Similarly here, it's been modeled as an asparagine, but Strudel tells you, well, it's either a proline or perhaps a glutamate, where you don't see the entire side chain, and it turns out to be indeed a proline. An asparagine has been modeled here, whereas the Strudel says, well, it looks more like a histidine, a phenylalanine, or a tyrosine, and it turns out to be a phenylalanine in the correct model. So this is a very nice way to potentially detect register errors and to fix them in Chimera. So the plugin works well with Isolde, so you can go ahead and start rebuilding your model straight away. So finally, I'd like to acknowledge my colleagues, Ardan, Samir, Peter and Garib, with whom we collaborate. Xue Wang has developed the validation pipeline and the validation analysis pages for EM. André Strate has developed 3D Strudel. The validation reports, there's a bunch of people on the EMDB side that have worked hard to implement all the EM specific parts into the wwPDB validation reports together with the OneDep team. Our EM Validation network also includes Elena, Maya, Martin and Ellen, in addition to Peter and Garib and Ardan and myself. We thank the wwPDB PIs and their teams for organizing the meetings and of course developing the software and annotating all the structures, and also the developers of all these packages that we are using in the validation analysis pages and or the 3D Strudel, and of course everyone who has deposited maps and models in PDB and EMDB. And I'd like to thank you for your attention, and I'll be happy to answer questions.